Okay, please, can you confirm you can see my screen?
Yes, yes, you can see your screen.
Okay. All right. So this evening, we'll be looking at everything polymerase chain reaction. Be looking at what PCR is. How it works. We're introducing you to the basic principle of PCR, the components of PCR reaction. That is, if you would be carrying out a PCR reaction, what are those things that you need? What are those components that would make up a PCR reaction? Then we also have the PCR thermocycle.
Hello, Olayinka. Can we all hear her? Or is it my network that is acting up? Can we hear her?
No, ma. I think it's a network.
Hey, please, let this not happen again. I mean, we had this same issue last week. Really, like, okay, let me try to reach out to her from here.
Then we conclude and take questions. But during
Oh,
the
we
course of
couldn't hear you for a bit, but yes, we can hear you now.
Oh, okay. Let me switch network. Let me
Oh,
switch
okay,
network.
okay.
Yeah. Okay, so I always love an interactive class, an interactive section. So during the course of the training, if you have any question. So I would take questions during the course of training and also when we are done. So I'll be starting now. Christiana, please can you confirm you can hear me clearly and you can also see my screen?
Yes, yes, we can hear you clearly and we can see your screen.
Okay. So PCR is, it is one of the most important, let me use one of the most important breakthrough in the field of molecular biology, because with PCR, with PCR, you can take a DNA sample and then produce millions of copies of that particular DNA. We all know we have four different biomolecules. We have the nucleic acid, we have carbohydrates, we have proteins, we have lipids. So for nucleic acids, we have DNA and we have RNA, and we know that DNA is the one responsible for the transmission of genetic information. So DNA is very, very, very important, and because of how important the DNA is in the body, a lot of scientists always a lot of scientists are always studying more about the DNA, studying more about the gene, which is a specific segment, of the DNA, because our DNA is composed of more than 20,000 genes, and each of these genes are coding. They are coding for a functional product. So let's say you want to study a particular gene. Let's say, for example, you want to study more about the gene responsible for producing insulin hormone. In order to do that, you would have to extract. You start with extracting you extract your DNA sample. Then after extracting the DNA sample. The next thing would be to carry out PCR reaction, because PCR would help you produce multiple copies of that gene. And then with that, it is easier to work with multiple copies of a particular thing compared to when you just have a copy. So PCR, polymerase chain reaction. So I'll be using PCR and polymerase chain reaction. I'll be using both interchangeably. So PCR is a molecular technique used to amplify a specific segment of DNA in a test tube, producing millions of copies from a small starting amount. Let's say you are interested in a gene that is causing a particular disease. So the first thing you would have to do is to carry out a PCR reaction so that you get more copies of that gene. So the simple thing PCR is doing is that it is helping to produce multiple copies. It's just like you have a book, you have a particular page of a book, you have a particular page, and then you want to make multiple copies. You want to make millions or billions of that particular page. Once you make the copy, people can do so many things with the different copies that have been made compared to just having a single copy of a book. So PCR was developed by Dr. 𝘄𝗲𝗁𝗵 in 1983, and then he later received a Nobel Prize in chemistry for his invention of the PCR technique. So this is the gentleman that helped with the invention of PCR. Before PCR was invented, what scientists usually do when they want to study a particular gene is that they would first isolate the cell that they can find that particular gene. After isolating the cell, they would now culture. It's like growing the cell in the lab, and that is going to take days. It's going to take days. Aside from taking days, it's going to take a lot of resources. But then with the invention of PCR, you don't have to wait days to make multiple copies, to make photocopies of a particular gene. So why is PCR important? It is very, very important for rapid DNA amplification. With PCR, you can take a DNA sample. Once you take a DNA sample, you do all the necessary setup, and then in less than 2 hours, you have multiple copies of your gene, and then you can now go ahead to do whatever you like with. You can go ahead to do other downstream processes with that DNA that has been amplified. Example of downstream processes that I can carry out can include sequencing, knowing the exact nucleotide base pair, knowing the exact nucleotide sequence in that particular DNA. Aside from that, it is highly sensitive and specific. With PCR, you are sure that you are working with the right. You are sure that you are working with the right gene. Like I mentioned earlier, the DNA contains so many genes, and then the genes are like segments. They're like segments on the DNA. So if you are probably using another method that is not as sensitive as PCR, you might end up working with another gene. It's just like you are building a house, and then you went to build that house on another man's land. You can imagine what would happen building your house on another person's land. Also, it is used across medicine, research, forensics, agriculture, and it is used in so many fields. It is not only limited to research. PCR is used in medicine. It is used in forensics. For example, in a crime scene, if you want, you can get a DNA sample, and then for crime scene investigation, the amount of DNA you would be getting from a crime scene is obviously going to be very, very small. It might just be the DNA you are getting from a strand of hair. So with PCR, you would be able to like produce multiple copies. You'll be able to produce multiple copies of that gene, and then that way you can easily identify the person that has that DNA. Then it is the foundation for modern genetic technologies like COVID-19 testing, cancer mutation detection. This COVID-19 testing is especially very, 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 very important because the field of molecular biology in Nigeria can say that it became more well known during COVID-19. Then it was during that COVID-19 period that a lot of hospitals, a lot of teaching hospitals, they acquired PCR machine, because with PCR machine, COVID-19 testing was, it was very much possible, and it was much easier than sending the samples outside of the country. So PCR is important for DNA amplification. It is highly sensitive and specific. It is used in different fields, and then it is the foundation for modern genetic
Hi,
technology.
Olayinka. Can you hear me?
Yes, I can hear you.
Yeah, okay, so we can actually hear you and we can see your screen, but I don't know if you've been changing slides because the slide is not changing here. Oh, now it is. Now it changed. For that period we were still on agenda, but now we are together. Why is PCR important? Yes, we are together now. Thank you.
Okay, I'm coming. Let me try to share the entire
Entire
screen. I
screen. Okay, okay.
feel that would work better. Okay, can you see it now? I'm on why is
Yeah.
PCR
Why is
important?
PCR important? Yes.
Okay, all right. So, is there any question so far from the introduction to PCR? Is there any question before we proceed?
Do we have any question? Anybody? If you have a question, you can raise your hand. I think
Can
we
I?
are good. Yes, you
All
can
right,
continue.
right. Okay, so PCR, this image here is just like giving us a summary of what PCR is doing. So, PCR, for you to work with PCR, you need your DNA of interest that you'd be extracting from your sample. You're probably getting your DNA from, let's say, you're getting it from maybe saliva, from blood, from spit, from stool, from soil, from air. You can get DNA from any can get it from anything. And thank God for science. Now we have a lot of extraction kits that have made extracting DNA from any sample. They've made it very, very easy. So you start with your DNA. You'd have extracted your DNA, you'd have purified your DNA to be sure that there are no impurities like proteins or other things that could interfere with the process. So you have your DNA. So once you get your DNA, we want to make copies. So we are starting with one DNA, and then from one DNA we are producing millions or even billions of that particular DNA. So what is happening? The first in the first cycle we are multiplying it, just from one sample we are producing 2 DNA. Then in the second cycle it is going to be multiplied, 2 times 2 4. Then in the third cycle 4 times 2 8. Then in the fourth cycle 8 times 2 16. In the fourth cycle 16 times 2, and then it would continue to increase like that, and before you know it, you would have millions of DNA that you can easily work with, but then this process, it has some basics that it has to follow. There are some basic steps that has to be followed before the millions of copies can be generated. So now we're looking at the basic principles of PCR, the basic principles of PCR. So PCR is a very very simple is a very simple technique. It is very 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 simple. When I say it is simple, trust me, it is simple, and then it is something anyone can carry out once they have the protocol, they have the procedure. So PCR follows three main steps. It follows three main steps, the denaturation, the annealing, and the extension. So it is these three main steps that would be repeated in each cycle. So in the first cycle, we have the denaturation, the annealing, and the extension. Then once the first cycle is done, the product from the first cycle will proceed into the second cycle like we have here. Then they would also, in the second cycle, the products would also undergo denaturation, annealing, and extension. Then after that, it would proceed to the next cycle, and then it would continue like that until the number of cycles you have set is reached. So for the principle of DNA, we have three main steps, like I've mentioned. We have the denaturation, the annealing, and the extension. So this denaturation, it is usually the first step. It is usually the first step because the DNA is a double-stranded structure. This that is in the background is the structure of DNA. The structure of DNA, it is double stranded, like two strands that are wound together, like a spiral, a spiral staircase. A spiral staircase. So the first thing in PCR is that we have to denature. By denaturing, I mean we are separating the two strands. And what we want is, we want the two strands to be separated. It's just like losing twist. For ladies in the house, there is braid, there is twist. Twist is just like two. So when we want to lose twist, by the time we are done losing our twist, we have like the two strands separated. So in denaturation, what is happening is that the two strands of DNA are being separated, and then initially it is hydrogen bonds that is holding these base pairs together. By base pairs, and I'm talking about adenine, thymine, guanine, and cytosine. In normal base pair in DNA, A would always pair with C, and G would always pair with C. A to T and G to C. So by denaturation, we are separating the two strands. We are breaking the hydrogen bond that is holding that adenine and thymine together, or the hydrogen bond that is holding guanine and cytosine together. And it is usually done at high temperature of about 94 to 95 degree Celsius. It is done at high temperature. This denaturation is just like melting your chocolate. If you put chocolate in your pot, in your fry pan, and you eat it to a very high temperature, it's going to melt. So in denaturation, we also, like you can say, we are melting. That is, we are separating the double stranded structure of DNA into single strands. You can see here, though it's not too clear, we can see adenine, thymine, adenine, cytosine, guanine. We want to like separate, we are breaking that thing, holding them together, which is hydrogen bond. So by the time the hydrogen bond is broken by that high temperature, we now have two strands of DNA. This is one strand and this is another strand. And we know that DNA is double stranded, and it has two strands running in anti-parallel direction. One is running in 5 prime to 3 prime, while the other one is running in 3 prime to 5 prime direction. So after denaturation, the next thing is annealing. So this annealing is usually done at 50 to 68 degrees Celsius. So it can be maybe 52, 55. It's usually within this range, depending on the primer design, depending on the primer you are using. So in annealing, what is happening is that the annealing step is helping to identify that gene of interest. Is helping to identify the gene of interest. In DNA samples, more than there are more than 20,000 genes. Just like you want to build you want to build a house, and then before you start building your house, you need to know your own land. Let's say there are about more than like 30,000 land. So you need to know which one is your own land out of all the 30,000 land, or even more, that are like very very close to your own land. So the annealing step, what the annealing step is helping to do is that primers would bind to the separated strand. Then these primers are like the one helping to identify the land that you want to build on. So they are the ones binding to the gene of interest, to that gene that we are particular about, the ones that would help us to identify that gene. So we have two types of primers. We have the forward primer, and then we also have the reverse primer. So they are both working on the two strands, forward primer on one strand, then reverse primer on the other strand. Then once the annealing step is done, the next step would now be the extension step. The next step would be the extension step. So we can say once you want to build a house, once you've identified your land, you start the foundation. Then after you've done the foundation, then bricklayers will now they'll start molding blocks one after the other until you get the structure that you want. So in the extension step, the DNase polymerase is like that bricklayer. It is an enzyme. It would now come and start adding nucleotide bases. It will start adding nucleotide bases to the 3 prime hydroxyl end of the primer, and then to start adding nucleotide bases one after the other, the new DNA, helping to build the new DNA strand. Because remember, initially we started with one DNA that is double stranded. This is the DNA that is double stranded. We carry out the denaturation step, which is helping to separate the two strands. So the denaturation step would separate the two strands. Once the DNA has been separated, the primers will now bind. They would bind to the region, marking. They will bind close to the region, marking the beginning and the end of the gene of interest. They'll bind close to that region, and then once the primers bind, DNA polymerase, the next thing DNA polymerase would now do is to extend. It would extend the chain and help to completely build the DNA strand of the gene of interest. And then at the end of the extension step, we are now going to have two DNA molecules, two DNA. Remember, we're starting with one, but at the end we are having two DNA molecules. So that is for the first cycle. So in each cycle, we have that denaturation, we have annealing, and we have extension. So that's these three steps are like the main thing occurring in a PCR reaction. So once you know the denaturation, the annealing, and the extension, you know all the different types of PCR that would be looking at. They also carry out this, they work based on the basic principle of these PCR. It's just that they are modified in one way or the other, but they are, in everything you will see denaturation, annealing, and extension step. So now that we are done with the basic principles of PCR, the next thing is to look at the component of PCR reaction. Component of PCR reaction. That is, if you want to carry out the PCR reaction, I already know that you are carrying out this denaturation and in an extension step. We've been able to establish that what are the different things that would make up that PCR reaction. So the first thing is your template DNA. Template DNA. You want to work on. So it's just like you want to cook rice. You want to cook fried rice. You need your ingredients. Yes. So we can say the components are like the different ingredients that would make up a PCR reaction. So you need your template DNA. Template DNA. It is this template DNA that would carry the gene of interest. No, without template DNA, PCR reaction cannot hold because there is nothing, there's no gene of interest to work with. So it is the template DNA. It contains the target region to be identified. It contains the target region to be identified. So, different types of DNA can be used, like the genomic DNA, the complementary DNA that can be synthesized from if you are working with RNA samples, it has to be converted to complementary DNA through the process of reverse transcription. Then we have plasmid, extra chromosomal material that can be found in prokaryotes. Then, another thing you need to take note of is that the template DNA must be clean and of good quality. It must be clean and of good quality. Just like you want to cook jollof rice, and then the rice you are working with is a rice that has stone. Now I don't want to know the chef that cooked the food. No, if the rice was not properly washed, it was not properly cleaned, there's no way you would enjoy that rice, because as you're carrying one spoon, you're eating stone. So you would even be discouraged. You'll be discouraged from eating the rice. So, and a PCR reaction, the template DNA, which is like a very, very important factor, it must be clean and of good quality. If it is not clean, it can affect the integrity of your result. At the end of the PCR reaction, you may get a negative result that the gene of interest is not there. And it's not that the gene of interest is not there. It's probably because the DNA sample has been degraded or something has happened, or it is not clean. So we need template DNA. So aside from template DNA, we also need primers. We need primers. So these primers are short single-stranded DNA sequences. They contain about 18 to 25 nucleotides. They contain 18 to 25 nucleotides, and what they are doing is that they are helping to bind to the complementary sequences that are flanking, that are like helping to mark that helping to know that gene of interest region. And they also define the region to be amplified, because there are so many regions in the DNA structure. There are so many regions. So to ensure that you are not amplifying the wrong region, you have a textbook. The page you are supposed to photocopy is, let's say, page 15, but due to error you are photocopying page 16, when you don't even need page 16, and you've made like millions of copies. The millions of copies amplified, and it must be specific. It must be specific to the gene, to the nucleotide it must have a complementary base pair only to the sequence, to the gene of interest in the DNA. So this bind exokinase really want to work with. So the primers, primers must be specific, and we have we have primer and the reverse primer. So the default primer is binding to the template strand. It is binding to the template strand, while the reverse primer is binding to the coding strand. It is binding to the coding strand, and they are pointing in same direction. Okay. So we have it here. This is the forward primer, 3 prime to 5 prime, and the 5 prime to 3 prime. The one binding to the 5 prime to 3 prime is the reverse primer, forward and reverse primer. Then we need DNA polymerase. They're like the bricklayer. It is one that is helping to synthesize the new DNA strand. It is helping to synthesize the new DNA strand, and it works best at 72 degrees. So an example of a DNA polymerase that is commonly used is this TAC DNA polymerase. It is a high fidelity enzyme. By high fidelity, it means that it rarely introduces the wrong nucleotide base pair. It rarely introduces the wrong nucleotide base pair, so it has high fidelity. And then another thing we need is the
Okay, I'm
NTPs,
busy.
which
Come
are
back
the
later.
oxynucleotide triphosphate. So the NTPs, they are like the blocks. You want to build a wall, you need blocks to build the wall. So they are like the blocks that are used to build the new DNA strand, because the DNA polymerase would be adding, it is adding this,
Yeah,
what it is doing, it is adding this building block
thank
one
you.
after the other. If the nucleotide base on the template strand is A, is going to add T as the complementary base pair. If it is T, is going to add A. If it is C, DNA polymerase would add G. If it is G, it would add C. So all these building blocks, adenine, thymine, guanine, and cytosine, they must be present. They must be present. Just like if you are building a house, you need blocks. So they are the blocks that will be used to build the new DNA. Then we also have reaction buffer. So this reaction buffer, it helps to maintain optimal pH and salt conditions. It helps to maintain optimal pH and salt conditions. It ensures that the DNA polymerase is working at an optimum it provides an optimum condition. Just like we have different some people now, they might function well in cold weather, some people function well in hot weather, some people function well at night, some people function well in the morning. So the reaction buffer is just helping to provide that optimum condition to ensure that the reaction is going well. We also have the magnesium chloride. So the magnesium chloride, what it is doing, magnesium is a cofactor for DNA polymerase. DNA polymerase is an enzyme, and we know that enzymes help to increase reaction rate. They help to increase reaction rate. So we have some enzymes that for them to be fully functional, for them to be holo enzyme, they require a non-protein part. DNA polymerase also require a non-protein part to be fully functional, and that non-protein part is this magnesium. So it affects the enzyme activity, primer binding, and fidelity. Then another thing that is necessary is nuclease-free water. In some cases, you might have extracted your DNA and you don't have enough sample. Nuclease-free water can be used to make up that volume. Maybe you need 40 microliter, you might need to add like 2 microliter of nucleus free water. You can't use normal water, because the normal water you are using it can be a contaminated water that would contain DNA that can affect your reaction. So we use nucleus, by nucleus free water. It doesn't contain DNA, or it doesn't contain RNA. So it doesn't contain DNA or RNA. Aside from that, it must be free from RNAs and DNAs, to ensure that it is not going to degrade the template, the DNA template, because DNAs can break down, just like protease would break down protein. DNAs can also break down protein. So ensure that water is nucleus free water to prevent degrading the DNA template. So these are the components, and one more component that we need is the thermocycler. So the thermocycler is the one helping to carry out the reaction. Once you get your reaction mixes, you add everything to a tube, you place it into your thermocycler. So once you place it in your thermocycler, you set the denaturation, the annealing, and the extension temperature. So it is the thermocycler that would do it. Without thermocycler, you would mix everything together. After mixing everything together, then you take it to maybe your water bath or your water heater, heat it to 95. Once you heat it, you bring it out, cool it to 55. Then you you cool it to 55, ensure primers bind. Then you now take it to increase the temperature again. So doing this process manually is very, very, is tedious, and you can even miss out, because imagine you are receiving a call during the process and you don't even know when it reached maybe 72, and what you need is 72. It can affect your result. But with a thermocycler, you don't need to do it manually. So a thermocycler would help with it. So you need your DNA sample, your primers, forward and reverse primers, your nucleotides, you need your DNA polymerase, you need your buffer, you need your magnesium chloride. You mix everything together in a tube and you add it to your thermocycler. So once you add it to your thermocycler, it goes through the process of the denaturation, annealing, and extension in repeated cycles. If you've set 30 cycles, it will carry out this step. It will carry it out 30 times until it reaches the last cycle which you have set. So is there any question so far? Can we pause a little bit? Is there any question?
Okay, I think Victoria has a question. Victoria, you raised your hand.
Yes.
You can ask
Good
your
evening,
question.
ma.
Good
Okay,
evening.
thank you. Yes, so when we were talking about the primers, I just wanted to understand from the annealing and the extension, those two phase. Like when the primer binds to the strand that has been denatured, right? That
Okay.
means the extension phase will now continue. I don't know. Like the primer will bind to the, maybe we want to get insulin now. The primer binds to that insulin gene, right?
Yes, it's
Or
going to bind to the regions that are marking that insulin gene. So that is where it is going to bind. No, that's where it is bind to. And then once the primer bind, it would add like short nucleotide sequence. So you now have like 18 to 25 nucleotide. Then after that, the DNA polymerase is now the one that would synthesize the strand completely to have a full double-stranded DNA.
for insulin. I
Yes,
don't.
for insulin, to complete the synthesis of the insulin gene. Let me use this.
Okay.
So let's say
Okay.
this is our insulin gene. So let's say this is our insulin gene. What the primer would do is that it would bind to, it would bind. Once it binds, it contains like short nucleotide sequence. So once it binds, the DNA polymerase is now the one that would completely help to synthesize that strand, that would synthesize the new strand. And then after the extension step, we now have a fully synthesized insulin gene.
Okay, thank you, ma. I get now. Thanks so much, ma.
All right, you're welcome. So is there any other question?
All right, James is raising his hand.
Okay.
James, you can ask your question.
Good evening.
Yeah, good evening.
My question is, I think during the course of your class, you mentioned only polymerase as the enzymes used during PCR. I want to know if there are also other enzymes used, and if yes, um, when are they added?
Okay, so the only enzyme we are using in PCR is the DNA polymerase.
Okay.
Yes, that's the only enzyme that we need, DNA polymerase. But we now have different types of DNA polymerase. So the TAC one is like a very, very common, it is a very common one. We also have PFU DNA polymerase. This one is usually used when you want to synthesize. Okay, so I said tag DNA polymerase is the only one. So it would be the only enzyme depending on the type of reaction you are carrying. If you are working with RNA samples and then with RNA, you have to convert RNA to cDNA. You have to convert it to complementary DNA. To convert it with complementary DNA, you would be needing a reverse transcriptase enzyme. So that's also another enzyme you can use, but that would be if you are working with RNA, not for DNA.
James, did you get that?
Yes, I did.
All right.
Okay. So is there any other question? Okay, I think we can continue.
Yeah, yeah, we can continue.
Okay, so we've been able to look at introduction to PCR, the basic principles of PCR. We've also looked at the components of PCR. So now PCR thermocycling steps, I've mentioned this. Once you are done with your reaction setup, you add your reaction mix to the thermocycler, and the thermocycler would run in cycle, and each cycle would contain denaturation, annealing, and extension. So the denaturation is where you are separating the strand, where you are separating the DNA strand. So after separating the DNA strand, the primers would anneal to the separated strand, then followed by extension, and the cycle would be repeated. So now I want to look at the different types of PCR. So the simplest type of PCR, I would say, is the conventional PCR. The conventional PCR. So this conventional PCR it is very, very, very easy. It is very, very cheap. It is cheap. It is not expensive. But one thing is that it is qualitative and not quantitative. So once you are done with the PCR, you are done with the thermocycline, you now take that product of thermocycline, you now like run it on gel electrophoresis. You're running it on gel electrophoresis. So it is now gel electrophoresis that will tell you that your gene of interest is present or your gene of interest is absent. So for conventional PCR, it is a type of endpoint PCR. That is, it is an endpoint PCR. You only know your result after the whole analysis is done, after the whole thermocycline step, and it is qualitative. It would only tell you, is your gene of interest present, yes or no. It won't give you the quantity, that is the amount. Maybe it's 3,000 copies per microliter of DNA, or 1,000, or 1. It won't give you any number. It would only tell you if it is present, and you need gel electrophoresis. I believe you guys must have done gel electrophoresis, but if you've not, it involves separating them on maybe agarose gel or any type of gel, depending on the type of analysis you want to run. And then you also, once you are running the analysis, you also load DNA ladders. You load DNA ladders. So the DNA ladder it would contain that your DNA of interest. So let's say we are interested in, I would also use insulin, because I know a lot of us know insulin, or we know glucagon, something that we all know. So let's say we are interested in insulin gene. Let's say this is insulin gene. Let's say this is insulin gene. We already know that if it is, this here is representing a DNA, this here is representing a DNA. So they're all representing DNA, or let me say they're representing a specific gene. They're representing a specific gene. So if our DNA is on that same line, we can say that, okay, yes, our DNA is present. Our DNA is present. Or let's say we know that our DNA, if present, is supposed to be somewhere around here. It's supposed to be somewhere around here. But now that we are not seeing anything here, we can say that, okay, in this particular sample, the gene of interest is not present. So this conventional PCR, it requires running gel electrophoresis after the amplification to help detect if your interest is present or not. Then we have the real-time PCR. The real-time PCR. So the real-time PCR, aside from primer, it uses something that is called probe or dye. The probe is like a tag, and it's like a tag that would fluoresce once the gene of interest is present. Once the gene of interest is present, it's going to like glow to produce a light that can be captured. So real-time PCR detects DNA in real time using fluorescent dyes or probes. And then for this real-time PCR, you'll be seeing as the number of cycles is increasing. You'll be seeing that, okay, there's increase in fluorescence intensity. So the increase in fluorescence intensity is helping you to know that, okay, yes, as the number of cycles are increasing, the number of gene is also increasing. So this real-time PCR quantifies gene. It quantifies, it gives you an idea of how much your gene is. It uses a standard. So it would use that standard as a reference to use it as a reference to tell you the number of DNA in the sample. And then this real-time PCR is often used in diagnosis. We also have reverse transcriptase PCR. So this one is if you are working with RNA samples, and you would usually see RNA in viruses, for example, COVID-19, or you have something to do with HIV. You would always have to carry out reverse transcriptase. You first extract your RNA. After extracting your RNA, you carry out reverse transcription to convert that RNA to complementary DNA. It is now that complementary DNA that would be amplified and detected. Then we have digital PCR. We have digital PCR. So let me go back a little bit. We have like different generations for different ages. We have the baby boomers, we have the Gen X, Gen Z, Gen Alpha, Gen Beta. So just like we have different generations for different ages, we also have different generations for PCR. We have different generations for PCR. So the first generation is this conventional PCR. So the first type of PCR that was produced, that was discovered, is this conventional PCR. Then from conventional PCR, we moved to real-time PCR. So a lot of people, a lot of lab are currently using this real-time PCR. Then we now have the third generation PCR, which is the latest generation PCR, and that is the digital PCR. So the digital PCR is the third generation, the latest generation PCR. So in digital PCR, what digital PCR is doing is that it is going to take a sample. You want to identify a gene in so many samples. Let's say you're working with cancer, you're working with a cancer, let's say BRCA mutation. You want to identify that BRCA mutation in, let's say, 10 sample. You have 10 patients that you want to test for that BRCA mutation. So what digital PCR is doing is that it is going to, for sample one, it will take sample one, partition it will divide that sample one into so many wells, let's say like 8,500 wells or 26,000 wells, depending on the type that you want to use. So once it divides it, it will if it contains the gene of interest or not. So for sample one, it will partition sample one into 26,000 partitions, and in each of the 26,000 partition, a PCR reaction is going on. By PCR, I mean the denaturation, the annealing, and the extension step is going on in those nano wells. For sample two, is going to partition sample two into 26K partition. Sample three, same thing. So it's going to partition all the samples into different nano wells, and in each of those partitions, it would now like check if the sample of interest is present, or it will check if it is present or not. And after checking if it is present or not, it can now give you the exact quantity of DNA in that sample. It will give you the exact quantity without the need for standard curve. For real-time PCR, it requires a standard curve, but for digital PCR, it doesn't require a standard curve. And digital PCR is extremely sensitive. It is more sensitive than real-time PCR. So it's just, let me use an analogy. You have a if you drop you drop an ink to a cup of water, and you are dropping the same ink, that same drop of ink, you are dropping it into a big drum of water. You would easily detect that ink in a drop of water compared to dictating it in a big drum of water. So what digital PCR is doing is that it is helping to increase the effective concentration of the sample. It is increasing the effective concentration of the sample. That way, it is easier to detect if your gene of interest is present or not. And then we have multiplex PCR. For multiplex PCR, you can determine more than one target in one reaction. We have different species of Plasmodium. We have different species. We have Plasmodium malariae, Falciparum, ovale, vivax. We have like more than 5 different species. So for those different species, you want to determine them in a sample, in a reaction. So for you to carry out this multiplex PCR, you would need the primer. You need primer for the different variants. You need primer. If we have like 5 variants, you will need 5 different forward and reverse primer that would bind to each of the gene. If you are testing for mutation, you want to check a wild type and a mutant gene, you know that you need to first you need primer that would help you to determine that mutant gene. You would need a primer that would help you to identify that original gene without mutation. So for multiplex PCR, you have more than one target that you want to determine, that you want to identify, that you want to amplify in a reaction. So it saves time, it saves time and resources. And this one is usually used in pathogen panels, which is why I used plasmodium example, for malaria testing. It is usually used for pathogen panels or genetic screening. And then we have the nested PCR. So in nested PCR, you are carrying out a two-step PCR reaction to improve sensitivity. You first carry out the first reaction. The first reaction would maybe you have 35 cycles, it would undergo those 35 cycles. And then once you are done, the product you've gotten from that first PCR will now undergo another set of PCR. It's like a two-factor authentication something. So it helps to reduce non-specific amplification. So those are the types of PCR. So now, what are the different applications of PCR? We've mentioned PCR. What is, why is PCR very, very important? What is it used for in different fields? So we have clinical diagnostics, in infectious disease. And a very good example, God so good, is COVID-19. So COVID-19 testing is possible with PCR. You collect the DNA sample, maybe from the throat or from the nose, using nose swab or throat swab. After that, you extract the DNA from that swab, then you run your PCR reaction. So if you get if there is amplification, it means that the person has COVID-19. The patient or the individual is positive for COVID-19. But if there is no amplification, if at the end of the process there is no amplification, it means that person is negative, doesn't have COVID-19. It is also used for cancer mutation detection. With the advent of PCR reaction, cancer testing is even much more easier now, because you get the circulating cell-free DNA sample. Then after getting the circulating cell-free DNA sample, you run the PCR reaction to test for mutations. So if a person has, let's say, EGFR mutation or KRAS, these mutations can be, they can be used to like test for different type of cancer, maybe lung cancer, liver cancer. So if an individual has the mutation, you can say that, okay, yes, the person has a specific type of cancer. It is also used for genetic disorder, which an example is sickle cell. Aside from clinical diagnostics, it is also very, very important in research. A lot of people, for those that are in their final year or those that are not in final year, a lot of people would be using this PCR for their reaction and for their project. Some, even though it is very, very expensive, but they will still use it for their reaction. It can be used in gene expression studies, genotyping, mutation analysis. You want to check if there is mutation, if a particular gene, if the nucleotide sequence has mutation, to know the sequence of a gene. PCR reaction is very important in reactions like that. It's also important in forensic science, DNA fingerprint identify a person. It is important in paternity testing. If you want to carry out, you have to, PCR is very, very important. You have to run that DNA, check if there's amplification, also for crime scene analysis, and then it is important in agriculture and food safety. You know, we have a lot of genetically modified organisms now, a lot of genetically modified food, modified tomato, modified corn. There's even a corn with Bacillus thuringiensis gene. You know, the corn has pest resistance. It has a pest resistant gene. That way, pests would not be able to like destroy it. So, you know, the process of producing that genetical and those genetically modified organism usually involves PCR, because you have that gene that you want to insert into the plant, you want to insert into the organism. It is important you must have studied the gene very well, know study how it's how it reacts and all. And before you can do that, you need PCR to help you make many copies of those genes. So PCR is important in agriculture and food safety. It is also important in environmental monitoring. It is important in environmental monitoring. You know, by environmental monitoring, you can collect wastewater, you know, maybe from a female hostel, and then there is constant testing of that. There's constant testing of the wastewater, let's say for maybe for polio or something. You know, if you are testing and then there is no amplification, or probably the level is very low, and then there is a sudden spike in the level of that part, in the level of the pathogen, you know, it's like a cause for alarm. Let's say you are testing for COVID-19 in wastewater, or maybe monkeypox or anything. You know, if there is increase in the amplification or increase in concentration, you know, it would quickly raise an alarm that okay, maybe a pandemic or endemic or something wants to happen. That way, you know, the government can quickly act. So PCR is important in environmental monitoring. So what are the advances in PCR technology? As we are here teaching, you know, there are some people somewhere already thinking that what can I do to advance science, what can I do to advance PCR, what can I, so people are like, you know, there's science is constantly being improved on daily. So there are a lot of advances in PCR technology. So we now have fast and touchdown PCR. You know, before, PCR used to like take time, but now in less than 2 hours you would get your results. You know, you'll be done with the analysis. So there are some innovations, some improvements, which include, you know, an improvement include combining this um annealing and extension step together. For digital PCR, it combines annealing and extension step as one. So instead of doing annealing separately, extension separately, you know, new PCR technology combines this annealing and extension step. That way, it reduces the time that the reaction would take. So it is fast, you know, it is fast and more efficient. Digital PCR is also, it is the latest PCR technology. It can be used to detect, you know, very, very, very low, even as low as 1 copy per microliter of DNA in a sample. It is extremely sensitive. Also, we have a lot of high fidelity enzymes, you know, for tag polymerase that have been like improved on, that would correct errors if introduced during the process of extension. And then we also have portable PCR devices. You know, there was a lab I went to in inside Lut, and you know, when I saw their PCR machine, I was surprised. Like, it was very, 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 very small, something you can hold with just one finger. So there are a lot of portable PCR devices that make field work very, very easy, and point of care testing. So in conclusion, you know, PCR is a very powerful, a very important innovation in PCR, in molecular biology field, and it is a versatile tool for DNA amplification, and it relies on three different steps that are repeated in cycles. It relies on the denaturation, which will separate the two DNA strands, the annealing, which involves primers binding to the region flanking the gene of interest, and extension, which involve completely synthesizing the new strand. So PCR relies on denaturation, annealing, and extension, and we have different types of PCR, and the type of PCR you would use would depend on the type of work you are doing. For example, if you're working on cancer research, digital PCR is advisable. If you are working with HIV samples or RNA samples, you are obviously carrying out a reverse transcription PCR. So different type of PCR you would use depend on the kind of work you are doing, and the application is wide in so many fields, from diagnostics to research to agriculture to forensics to environmental monitoring. So I would say thanks to the inventor of PCR for the wonderful technology. So thank you guys so much.
Hmm.
So I'll take questions now.
Wow, wow, wow. I mean, this was a full class. Thank you so so much, Olayinka. I was how do I say it now? I was glued from beginning to the end. I like how that you took your time to break every step down. I mean, I know some of these things to some extent, but with this class, I can categorically say that no, my my knowledge of PCR has really been increased and amplified, right?
Yeah,
Just
it
like
has been
PCR is
amplified.
amplifying DNA. So
Gone
let's
from
just
one to
see.
millions.
Exactly, exactly. So let's see. Do we have questions? Anybody? Do we have any question from anybody? I think some persons joined along the line and they had to leave. However, for those of us that are still on the call, do we have any question? I actually don't have any. I don't think I have a question because you really took your time to explain every detail. So I really don't have a question. But let's see.
Like
Victoria, any question?
James, Taiwo, any
Yeah,
question?
Taiwo. I think Taiwo was on the call from beginning. Etima, do you have any question for us? In the absence of questions, do we have anything to say? Probably, or what's the new thing you learned or something? I mean, we just want to know, we want to be sure that you really learned something. So should I call on our names? Let me call. I'm going to start with Taiwo. Taiwo, what did you learn from tonight's class?